Hi guys, thanks for coming. I think over all the talks I've seen over the last few days, I think this goes without saying, but models form a pretty integral part in the way that we synthesize and understand the natural world. And beyond that, they also form a pretty integral part in how we forecast into future and usually quite novel conditions. However, quite often, the barrier to entry for some of these models, especially for early ecologists, can be quite high. And there seems to be, especially from a computational point of view, quite a few barriers with being able to jump into a modelling system and begin to run models. And it's not because ecologists can't learn this. Of course, we're ecologists, we can do everything. It's more that the time it takes to learn and synthesize these models, learn about the data and everything that surrounds it can take a lot of time. So I'm here mostly to talk to you today about the Biodiversity and Climate Change Virtual Laboratory. But before I delve into it, and I'll give you a bit of a demo of how the lab works. However, before I do that, I wanted to just touch on exactly what a virtual laboratory is. So virtual laboratories are formed around engaged research communities, and through that engagement with these communities, we build on existing research capabilities. That is, virtual laboratories don't pave the way. However, we aim to kind of help and synthesize the way that we currently do things. We support research workflows, and we provide easier access to data and analytical tools across a broad range of disciplines. And by doing that, we facilitate the sharing of data as well as encourage collaborative efforts, not only within institutions, but also across institutions. So to give you a few examples of these, there's the BCCVL, which I'll demo for you today. There's also things like the Genomics Virtual Laboratory, which basically removes the highly specialised IT component out of genomic work. There's the Virtual Geophysics Laboratory, where they make available geophysical tools and data and a heap of resources. There's also the heroically named MARVEL Laboratory, which stands for Marine Virtual Laboratory. There's the CVL, which is the Characterization Virtual Lab. There's the All Sky Virtual Laboratory, which I'm massively simplifying this, but it's pretty much a really awesome telescope that's on your computer. There's also the Climate Weather Science Lab. There's HONEY, which is the Humanities Networked Infrastructure Lab, which unites a whole heap of cultural data. And there's the Industrial Ecology Lab. So basically, this is just a suite of a few of the ones that are available here in Australia, but there's also virtual labs all over the world. And the aim of these labs is to make available computational resources to research disciplines. So back to the BCCVL. The BCCVL is a decision support and scenario planning tool that simplifies the process of biodiversity and climate change modelling. We're guided by a suite of experts, and it's been a massive collaborative effort to get this lab up and running and to continue it functioning today. So how does the lab work? Basically, we provide access to around 4.5 terabytes of data across environmental, climate, and biological disciplines. And by doing that, this data is available in the right format to be used in models. You can visualize it, you can interrogate it, it's all there ready for you to use. We also, and this is kind of the powerhouse of the lab, we connect to over 400 central processing units. So currently, computers, I guess my general laptop, correct me if I'm wrong if there's any IT people here, but I think current laptops are about 4 central processing units. So what happens in the lab, you submit your experiments, it bundles it up, sends it off into the cloud, sends it to these 400 central processing units, runs your models, and then shoots them back to you when they're ready. This frees up your laptop to do 100 other things because we're all very busy. And it means you still get all your results and can run your models. One of the really cool things about this, and just before I walked into this room, I ran a quick check on how we're running. And at the moment, to run one generalised linear model with climate and environmental layers and about 100,000 occurrence records, it takes 50 seconds to complete that model. Another thing is we do all the coding and script work for you. So the BCCVL is a Python-coded platform, and all our scripts run off R scripts. So we tap into Biomod2 and Dismo programs. So we've got 5 experiment types. We've got 2 primary experiments, species distribution model experiments and species trait experiments, which are currently under development. And these primary experiments feed into our secondary experiments. So these are climate change experiment, ensemble analyses, and also biodiverse experiments. In the interest of time, I'm going to mainly focus on species distribution models and climate change experiments today. So in the lab, we offer 17 algorithms that you can run. And again, a really cool function of this is you can go in and run all 17 at the one time to compare the outputs. And we've got the ability for you to be able to compare them side by side to have a look at the differences in these algorithms. This is a quick demo that I ran this morning. So I've logged into the lab. I'm searching for, I've gone into the datasets tab to make sure I've got all the data I want for my model. I'm looking at the current climate data. This is generated by JCU. This looks much prettier on my Mac, but still. So as you can see, you can visualize all the bioclim layers. You can zoom in, you can interrogate them. You can start to get an idea about how this data might affect your species. We've also got a bunch of environmental data in there. This is the MODIS FPAR time series data, again, which you can visualize, zoom in, interrogate, look at. We've also got other things like hydrology. At the moment, we're trying to get some land use data in there. This is the vast data set here. So all of these are ready to be used in the models. From here, you can go, we also import from the Atlas of Living Australia. It's a live feed we have to their database, so you can type in your species of interest, pull it into the lab. We recommend putting it into your computer, cleaning it, and then re-uploading it. So to run a model, you go to the species distribution modelling experiment, type in, or give your experiment a name. Select your species occurrence. You've got to work your way through about 6 tabs. So this is basically just a nice interface for what's happening behind the scenes with the R scripts. We can generate pseudo-absence points, or you can upload your own. You then select your climate and environmental data sets. Again, you can visualize them here if you want to sort of recall that. You select the variables that would be most suitable to your species. Hit next. This is a new feature we've just added in, which is geographical constraint modelling, which means you're able to train your data or your model to a given area and project within that. And then, of course, these are all our algorithms. So you can select 1 or 2. You can select them all. We've also got configuration options for each algorithm. Again, these are all set to default. So if you don't want to or don't know what you're doing, you can leave it. And as you get more and more familiar with models, you can start to really tweak these. This is what your experiment's results look like. So you get all your response curves, of course, your probability map, your ROC curves, all your evaluation statistics. It all comes back to you, sent to you in a nice little email. And of course, you can download all this to your computer. A neat function we have is the ability to overlay your probability maps, so overlay different algorithms, and also to compare the maps side by side. So you're able to, especially from a learning and teaching point of view, you're able to kind of display the differences between a geographical model and a machine learning model. And of course, you can compare the maps side by side as well. Whew. So this is a climate change experiment. You go in, you can select your species distribution model that you've just run. This is input into your climate change experiment. Select your emission scenarios, your global circulation models, the years you want to project into, as well as the resolution you want to run your model at. Select the resulting data and start your experiment. So this is a corroboree frog. That's the original distribution model on the left, and on the right is 2085 under the model, under the RCP 8.5, which is the business as usual. So that's a very quick example, and I'm running out of breath because I had to speak so fast. But essentially, admittedly, I've sped that up to be twice as fast. But if you imagine half that time, and that's how long it takes to run a species distribution model and a climate change projection under this system. Now, one of the important things is, of course, with great responsibility comes blah, 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 as Spider-Man says. But most importantly, by simplifying such a complicated modelling tool, we do open the doors for people to run perhaps poor models or look at them and interpret them poorly. So in a means to kind of combat that, we've started an online open course in species distribution modelling. And in this course, we have done a series of animations to try to inform the users about the data you need, the data qualities, what you need to think about indirect and direct effects on your species. We've also gone and looked at every single algorithm and how it works behind the scenes. So what it does with your data, how it treats your variables, whether there's interactions, whether it's additive. And we've tried to do it in a really sort of graphical, engaging manner, but that's still clear and concise. One of the things that many people know is some people or some modelers use a particular algorithm that they really like or something like that. This kind of opens the doors to being able to look at many different algorithms, try to understand the intricacies between them, and things like that. And of course, we've got a bunch of modules on your evaluation statistics, so how your model cross-validates between your data, how it uses all of that, or your contingency tables and all your evaluation statistics, and how you should interpret those. That's the list of all the modules in the course. That's coming out in March, and it's free to access. So in summary, the BCCVL and virtual labs in general are a really good stepping stone for understanding the concept of modelling without getting too bogged down in the coding or the data format side of things. They're really good as an educational tool for up-and-coming modelers. It does streamline, and especially for researchers and perhaps people that are already modelers and know what they're doing, it does streamline their research by connecting them to Australia's computational infrastructure. Next up for the BCCVL, we're currently in beta release, so we're working with our scientific advisory committee to get us into full production. It is still open and available for everyone to use. We're looking to be integrated into undergraduate and postgraduate courses. One of the things we're doing is refining our regional constraint modelling. So at the moment it's just a box, but we're looking to be able to refine it into EBRA regions and local council areas. We're also really close to releasing a multi-species modelling, so being able to upload one CSV file with 800 species and run a model on all of those and not crash your computer. And of course, integrating conservation planning tools like MarkSand and Zonation. We're also looking to integrate more international data sets, so we're about 2 weeks away from integrating GBIF. And of course, we're always open for data suggestions. So if there's any data that perhaps is useful to your models, we want to know about it because we're happy to put in the man hours to get it sort of ready. Thank you to all the partners and of course to Nectar who fund our project. Thank you very much.